So our next speaker is a selected speaker from the abstract. It's Adrien Dufour. He's from it's an abbreviation from the Animal Genetics and Integrative Biology unit of the INRAE in Jouan-Josas in France. And again, as soon as his slides are going to be on, he's going to talk about multi-omics analysis of pig pre-implantation embryos.
Hello everyone, I will talk to you about my PhD project, which is analysis of multiomics in the pig pre-implantation embryo. So why study the pig pre-implantation embryo? So the use of pluripotent stem cell in livestock species is still limited by technological limitation. And the optimized mode of obtaining pluripotent stem cell for rodent and humans are not well adapted for pigs species. So a working hypothesis is that in the pig and the cow, contrary to mice and humans, the implantation is much later, around 20 days after. The other differences is that the trophectoderm cell here that covers the epiblast, the pluripotent cells, progressively disappear from year 7 days. And the embryo will continue to elongate. So this particularity of pig and cow are not well documented. So the working hypothesis is that the paracrine regulation specific to the pig embryonic development are not taken into consideration for the establishment of pluripotent stem cell line, and that the embryonic pluripotency regulatory networks are not necessarily conserved in all the mammalian species. So we perform a single-cell RNAseq at different stages of the pig embryo development, from the early blastocyst to the ovoid blastocyst. From those same we also collect uterine fluid, and we performed mass spectrometry for the quantification and identification of the protein fluid. And lastly, we also done single-cell multiomics, which we can access to the region accessibility and transcriptomics for the same cell. So first result on single-cell RNAseq allow us to firstly identify that the early developmental stage are clearly different from the other stage of the embryo development. And from literature clonage, we also identify the different lineage of population, the trophectoderm here, the hypoblast, and the epiblast. We then search to characterize more deeply all different tree main lineage. And here you can see the characterization of the trophectoderm population. And we can observe that we identify a specific population of interleukin-1 expression. A second population of trophectoderm ARP2, that's a potentially stem cell population. And lastly, a population of trophectoderm MT that were apoptotic cells as they express marker of apoptosis. So that maybe correspond to the polar trophectoderm that I described previously, that has nomed the rubber layer, and that progressively disappear. as this population is specifically identified at the E9 stage, and it disappears immediately after. So then we try to identify module of regulation, regulon. So regulon is a composition of transcription factor and co-expressed gene with a common motif. So to do so, we use a package named scATAC-seq, which has been developed to identify this regulon in a single-cell dataset. And I also perform a meta-analysis using a human and pig datasets published in a pre-implantation embryo. And you can see on this heat map that we identify nearly 300 regulon, and we find the regulon known in the literature, like nanog for the pluripotency, also on the one for trophectoderm cells. And you can see that a lot of our transcription factor identified are also identified in pig, and less are identified in both pig and human, and in humans. So next, we try to identify possible cell communication between our different cell population in our datasets. So firstly, we can see on this core diagram, we are here the ligand sending gene, and here the receiving receptor. And in those results, firstly, you can see something that have been previously described in pig is the importance of the DAC-STAT pathway in the early embryo. As you can see, EL6R and EL6ST are receptor and ligand of that pathway. But we also identify a new interaction, like the integrin pathway with autocrine signal from the inner somas to the inner somal, and the paracrine signal from the trophectoderm to the inner somas. So we try then to see in the proteomic feed analysis, and we we got 28 samples from the same source as the single-cell RNAseq, and we can identify that the feed differentiates between the early stage and the late stage on this visualization. And we next try to, can we connect those proteomics data to our single-cell transcriptomic data. So we use a non-package from single-cell RNAseq, and we search for ligand and receptor interaction. So you can see here in yellow the ligands that correspond to our uterine threads, and in blue the receptor that come from trophectoderm or the epiblast. And that we have been found is firstly interaction between the pig endometrium and the pig embryo with EL1 RP and EL1 R1 that have been previously described. But we also underline the importance of the extracellular matrix components. As you can see, we found a lot of integrin, fibronectin, and collagens. And then we also identify the F-win pathway that have been described in the pluripotent mouse embryo. So Next, we will try to a little bit confirm the regulon that we identify previously. So to do so, we decide to adding a layer of information using the analysis of multiomics to a little bit strengthen our conclusions. So on those data, we have been provide 8 clusters of cells, and we characterize this cell based on literature knowledge. And you can see on the plot, firstly, the lecture of the read on the genome, the peak that result that have been identified, and here's the gene expression corresponding in the different clusters. And from these two information, we can infer a peak to transcription link that you can see here, as you can see the position of the gene here. And you can see that for this 4 populations that we assess to be the hypoblast, as 2 of them express GATA-cis, you can see here that they have a specific peak in this 4 population. And you can also see the same thing for DAPE2 for the trophectoderm, and Poussin-Keffin, you can see that the transcription level are high. So the newly developed scENIC approach, but for the multi-omics datasets, allowed us to add the open chromatin information. So you can see here the first group of activator. So when scENIC discover transcription factor and gene open chromatin in the same cells, and a gene expression of the gene of the regulon is also in the same cell. And here, one repressor, so when the transcription factor is expressed, the region is closed and the expression is shut down. And what we can also see is that in the blue to red, the level of region accessibility score, and in the side of the dots, the gene transcriptomic level activation. So you can see that we found 55% of similarity with previously single-cell RNAseq analysis. And we also find some literature non-transcription factor that we can't find in our previous approach, like CDX2 and PU5if1. And then we shall see if for this regulon, can we see footprints to confirm the presence of the transcription factor in those populations. For those who don't know what is footprints, in the scATAC-seq analysis, when the transposase will cut the region of open accessibility, if the transcription factor is still fixed to the DNA, the transposase cannot cut those part. So you can see that for the motif 4 nanog, in the particular population of the epiblast, we have a clear difference for footprint for this motif in those populations. And we can also see for GRHL2 that for the 2 populations of trophectoderm, there is a footprint difference with the other population. And we can also see that for FOSL2, that for the 4 different populations of hypoblast, the level of the footprint will be slightly different. So to conclude those analyses, it allowed us to identify new population of trophectoderm in the pig blastocyst. The identification of K-paracrine and autocrine interaction between the epiblast, the trophectoderm, and the uterine fin. And the multiomics allowed to refine or previously identified regulon and vector characterize them with regions accessibility and motif footprint. So thanks for the listening and thanks for the team that host this on our collaborators.
Thank you, Adrien. I think we have time for one question. Then I have a quick question. You went very quickly over your multi-omics data. What is the source of this data? Is this literature-based, or do you generate it yourself?
Yes, we generated on the pig embryo, not from the earliest stage because of the difficulty of the embryo at the stage because it's very...
But it's scATAC-seq data then, or is it...
It's multiomics, so we have scATAC-seq and transcriptomics.
Yes, exactly. Yes. Okay, but this is the source of the data. Okay. Thank you very much.